Welcome viewers. Amid the ongoing COVID-19 pandemic, the development and distribution of vaccines have become paramount in our collective efforts to combat this global health crisis. Vaccines have long been a powerful tool in preventing the spread of infectious diseases, and COVID-19 vaccines are no exception. In this video, we will delve into the different types of COVID-19 vaccines currently available, exploring their mechanisms, benefits, and limitations, and addressing common misconceptions. But before we dive into the specifics, let's first take a moment to understand the importance of vaccines in general. Vaccines are biological substances that help train our immune system to recognize and fight off harmful pathogens, such as viruses or bacteria. They typically contain weakened or inactivated parts of the pathogen, or sometimes just fragments of it, which triggers an immune response without causing the actual disease. This immune response allows our bodies to recognize and remember the pathogen so that if we encounter it again in the future, our immune system can mount a swift defense and prevent the disease from taking hold. Now, let's discuss why COVID-19 vaccines are so crucial in our fight against the pandemic. COVID-19, caused by the SARS-CoV-2 virus, has wreaked havoc on societies worldwide, leading to millions of cases and loss of life. Vaccines offer a glimmer of hope and a pathway towards curbing the spread of the virus. By achieving herd immunity through widespread vaccination, we can protect not only ourselves but also the most vulnerable members of our communities, including the elderly and those with underlying health conditions. Understanding the different types of COVID-19 vaccines is essential for making informed decisions about vaccination. So, without further ado, let's explore the various vaccine types. Traditional inactivated or attenuated vaccines. Traditional inactivated or attenuated vaccines have been used for decades and have a proven track record in preventing numerous diseases. These vaccines utilize either killed or weakened versions of the virus to stimulate an immune response. By introducing the inactivated or attenuated virus into the body, the immune system learns to recognize and respond to it, building immunity against future infections. Examples of traditional inactivated or attenuated vaccines include the polio vaccine, measles, mumps, rubella, MMR, and the seasonal flu vaccine. Despite their effectiveness, traditional vaccines have some limitations. In some cases, the inactivated or attenuated virus may not generate as strong of an immune response as desired, requiring multiple doses or booster shots to achieve optimal protection. Additionally, there is always a small risk, albeit rare, of the vaccine actually causing the disease it aims to prevent. 2. mRNA vaccines. mRNA vaccines, such as the Pfizer-BioNTech and Moderna vaccines, represent a groundbreaking advancement in vaccine technology. Instead of using a weakened or inactivated virus, mRNA vaccines utilize a small piece of the virus's genetic material, known as messenger RNA, mRNA. This mRNA provides instructions to our cells to produce a harmless piece of the virus, known as the spike protein. Once produced, our immune system recognizes this spike protein as foreign and mounts a response, producing antibodies and activating immune cells. This immune response provides protection against future encounters with the actual virus. One of the significant advantages of mRNA vaccines is their ability to be developed and manufactured quickly. This has been instrumental in the rapid response to the COVID-19 pandemic, allowing vaccines to be developed and tested in record time. Furthermore, mRNA vaccines do not contain live virus particles, eliminating the risk of causing the disease they aim to prevent. Despite their numerous benefits, mRNA vaccines have also been met with some concerns and misconceptions. One common misconception is that mRNA vaccines can alter our DNA. This is simply not true. mRNA vaccines do not enter the cell nucleus where our DNA resides, and they are rapidly broken down and degraded within the body. Another concern revolves around the short-term side effects reported by some individuals, such as fatigue, headache, or muscle pain. These side effects are generally mild and temporary, indicating that the immune system is responding to the vaccine. 4. Viral vector vaccines. Viral vector vaccines, such as the Oxford AstraZeneca and Johnson & Johnson vaccines, employ a different approach to stimulate an immune response. These vaccines use a harmless virus, known as a vector, to deliver a piece of the coronavirus's genetic material into our cells. Once inside the cell, this genetic material instructs our cells to produce the spike protein of the SARS-CoV-2 virus, triggering an immune response similar to mRNA vaccines. Viral vector vaccines offer several advantages, including their ability to induce a robust immune response and potentially provide longer-lasting immunity. Additionally, viral vector vaccines can be stored and transported at standard refrigeration temperatures, making their distribution and administration more feasible in certain settings. However, viral vector vaccines also present unique challenges. Some individuals may have pre-existing immunity to the viral vector used in the vaccine, potentially reducing its effectiveness. Additionally, rare cases of blood clotting disorders have been reported in individuals who received viral vector vaccines. While these cases are exceedingly rare, regulatory agencies and healthcare providers closely monitor and manage these risks. V-protein subunit vaccines. Protein subunit vaccines, like the Novavax vaccine, work by introducing harmless pieces of the virus, typically proteins, into our bodies. These proteins trigger an immune response, allowing our immune system to recognize and remember the virus in the future. Protein subunit vaccines can be produced using different methods, including recombinant DNA technology or growing the proteins in cell cultures. One significant advantage of protein subunit vaccines is their safety profile. As they do not contain live virus particles, there is no risk of causing the disease they aim to prevent. Additionally, protein subunit vaccines can be easily manufactured at large scales, making them suitable for widespread distribution. However, protein subunit vaccines may require the addition of adjuvants, substances that enhance the immune response, to achieve optimal efficacy. Adjuvants help stimulate a stronger immune response, ensuring long-lasting protection against the virus. Furthermore, the production of protein subunit vaccines can be complex and time-consuming, leading to delays in availability. In the rapidly evolving landscape of COVID-19 vaccine development, researchers are continually exploring new avenues and technologies to combat the virus. Current research developments include the investigation of multivalent protein subunit vaccines, which target multiple variants of the virus simultaneously, as well as the development of new adjuvants to enhance the immune response. In conclusion, the development and distribution of COVID-19 vaccines are crucial steps in our battle against the pandemic. Understanding the different vaccine types empowers us to make informed decisions about our health and vaccination choices. Traditional inactivated or attenuated vaccines, mRNA vaccines, viral vector vaccines, and protein subunit vaccines all play a vital role in our global vaccination efforts. Remember, it is essential to conduct thorough research and consult with healthcare professionals before making any decisions regarding your health or vaccination choices. Stay informed, stay safe, And let's work together to overcome this global health crisis.